Hi everyone, it's so awesome to be here. Thank you so much for the invite. I'm supposed to stay away from one of the speakers.
That one.
That one, okay, cool, so it doesn't reverberate. So I'm going to tell you a fun story about the soup that blew up Twitter. This was the title that Twitter actually voted on as the one to use, so democracy rules, right? I've been a microbiologist for 16 years, and yet the thing that I am most known for is the soup that rotted in the back of my fridge. This is not my fridge. This has way more fresh produce than my fridge does. I will be a college student forever, but it's similar to what it would look like. But this is not just a story about the science experiment in the back of my fridge. It is also a story about the joy that science can bring, the community that science can bring, and the education to the general public that science can bring, and how Twitter played a role. Twitter and a great community on there played a role in it. So this all started with a birthday tradition. My mom, every year for my birthday, lovingly makes this delicious beef soup that is her grandmother's recipe. And it warms me up both physically and it warms my heart up. You know, it's one of those I look forward to every single year, and she makes me like this giant vat of it. So I've got leftovers and fresh stuff in my fridge for a certain period of time. And so this past year, I was going in and like taking my leftovers, like ladling out, because I just had the pot in the back of the fridge, and I kept on like ladling and everything, and I had a little bit left over. And you know when it's been like a week and some, and then you're just like, okay, maybe not anymore. Maybe I won't go back into that anymore. Maybe some of you are better than me, but it's a thing. And so, you can't really see the soup as well up here. But so after a while, it was just forgotten in the back of the fridge, and a month goes by, and my fiance is like, okay, it's time to let's actually clean out the fridge and be like adults for a second. And so then I tweeted, okay, I'm outing myself here, but there was this forgotten beef soup in our fridge, and we just cleaned it out, and it was blue, like bright blue. I'm like, what the F contamination would make it blue, like bright blue? And even with all my years in micro, I'm just like not handling this well. I'm just like, And I should have known that, because my Twitter community is mostly microbiologists, that they would not let it slide. So we had people start chiming in. So Kat, she's not a scientist, but she managed to get adopted by the science Twitter community. But she was like, please tell me that you have a photo. And I was too busy gagging to try to get, I don't want to risk my phone or risk vomiting. I was like holding my breath the whole time. And she's like, come on, Elinne, what kind of scientist are you? You didn't even document your findings. And I'm like, oh, you hit the twinge. But a bunch of others were also saying, like, you gotta, come on, you gotta send us a photo. You can't just leave us like this. And I'm like, there are limits about what I'm willing to do for science, for sure. And then more and more people were like jumping in. I'm like, no, I am not going back into the trash. It is double bagged in there. I poured the soup out, like those like thick reusable like grocery bags. And so I double bagged those, tied it up, and threw it in the trash. You could tell I wasn't even typing well, how like distracted I was by this whole thing. But of course I should have known that my audience was like a group of microbiologists and would not let this slide. So after a little while, I was like, okay, I can't believe y'all talked me into going back into the trash. Warning if you have a squeamish stomach. It is bright blue. It's a little dim up there, but if you actually see like the full photo, you can go on my Twitter, it's on the pinned tweet, and it's like bright up there. And so then that's when Twitter erupted, because they're like, this is like microbiologist's dream, right? That's how penicillin was discovered, was from rotting food. So we have people like, you might have found the newest antibiotic, take a sample please. And I'm like, I'm not going back in a second time, you've got to be kidding me. And so then Helen was like, hit it with the black light and see if it glows. Like everyone's telling me, sample, do different things. I'm like, I am not going back into the trash a second time. And so then Michael chimes in with, hey, it's not our fault you didn't save a sample for testing. And I was like, oh, he knows how to like make my little stubborn like brat like come up and everything. So I was like, oh my God, no, you guys, this is gross. It's been sitting out there for a day now. You gotta be kidding me. So then in comes Sebastian, who's the second main player in this story. Sequence it, oh my God, I'll pay for the flow cell. That's the actual, like the part that you do the sequencing on. They can range from anywhere from like $600 to like $8,000 for sequencing, depending on how much that you want to sequence. And I'm like, I'm not going back in the trash. It's been sitting outside for a day. There's no way. And so he's, but the Azurans. And then Pedro chimes in and piles on. This would be the coolest paper idea of all times. Come on. And then I was like, they're going back and forth. I'm like, you guys, I'm like 43% tempted to go back into the trash. And Sebastian, he won't let up. He's like, honestly, even a little EP tube, like this little like plastic tubes you see, or even a Q-tip would suffice. Because nausea is temporary, glory is forever. And he's like, we can even call it Becket Blue, come on. And I'm like Sebastian. And then a few hours later, in comes, come on, it's not working. Can you hit the next slide? It's not working for some reason. There we go. I'm never forgiving you for this. So I take a photo. I literally, because I was at home and I'm like, I'm not going to lab to get a swab and coming back. I literally went upstairs to my bathroom, got a Q-tip, opened it up, dunked it in there, like it's all up on my arm and stuff, and then I threw it into a Tupperware that I had, and I'm like, I am absolutely never forgiving you for this. So Sebastian was like, yeah, let's do it, because his passion is to be able to isolate different blue genes so he could do some sort of plant bioengineering. We'll talk about that in a little bit. But he's always wanted to do blue. He wanted to make like a blue rose, things like that. So he's like really obsessed with this color. And so then we went back and forth, and he convinced me to go back in a third time so that we can get 8 swabs so we can do all the different tests and stuff. I'm like, that's the last time I'm going in. He goes, a true hero, the soup may rest in pieces. So I'd like to introduce Sebastian a little bit more. He is a self-taught, quote, amateur biologist with no formal training or degree. And so I put it in quotes because Sebastian is by no means an amateur. He operates a home lab and has been working in the plant bioengineering sector for 15 years. And he's such a brilliant person. Like he knows as much as like pretty much any professor that I know. And he's also like the purest human being that I've met. He just always wants to like make the world better and make everything around him better. And his research focuses on bridging the gap between research and education, working on tools and techniques that allow everyone to be able to participate in science. And so one example is he's engineered these safe bacterial strains that are all these different colors of the rainbow so that people can paint with bacteria to be able to learn different things about science, and as well as be able to use these in different types of experiments that many labs use as well. So Sebastian's awesome, and he's just like so eager. He's like, let's study this, right? So the investigation begins. But first, the cat's out of the bag. So my mom came across the thread, and I had to answer for the fact that I let her soup spoil. And secondly, she's worried that we'll figure out the recipe, right? I mean, I know the recipe, but I mean, I've been sworn to Croatian blood oath secrecy, right? So, first of all, I would like to go on record that my mom's cooking techniques are safe and really, really good. The contamination happened after, with me going in a lot, so it's the microbiologist's fault, not the Croatian mom's fault. But even though millions of people saw this whole saga and it blew up and people questioned my mom's cooking as a result of that, regardless of all this, what did she do? She wrote me a sonnet. So she wrote me a sonnet, Ode to Blue Soup, and she framed it and sent it to me in the mail. And I was like, okay, so now Sebastian and my mom are the two purest people that I know in the world. So, this works sometimes, but not always. There we go. So, now what's in it? So one thing that literally thousands of comments said were like, is there onion? Is there cabbage? Is there, well, red onion specifically, is there red wine? Is there garlic? Is there all these things? And I'm like, nope. I've had to like answer this 100 times. So since this is a great debate, no, there's not cabbage, beets, garlic, surprising, because we put this in everything when we cook, tomatoes, soy milk, any of this stuff. To a point, this got so out of hand that Liz had to make this meme for me so I could just share it. No, these are not in the blue soup. Because there are these class of compounds called anthocyanins, which, like if you ever see, like, you know when you have red cabbage, if you add vinegar, it turns pink, and if you add baking soda, it turns blue. And so you have these different compounds that could like seep into, like from these different veggies that could seep into and explain the blueness. But it didn't look that color, especially if you take a closer look at the soup. Come on. Next, come on. There we go. It's hard to see with the projector, but if you take a closer look, it looked bacterial. It just like to us who have been dealing in microbiology for so long, I mean, sorry for any mycologists on there, I didn't think it was fungal, because like the texture was a little bit different, and just the color and the turbidity just made me, Sebastian, I think, like this probably was bacterial. Also, there's other studies that had been done. Why is this not working? There we go. Other studies that had been done, like Parker, they shared a link, and others share the link as well, to the study I'll talk about in a little bit, that had found that soy milk and tofu and other people had found cheeses that had turned blue that were as a result of bacteria. So the study that they linked was Marina De Leon study, and with her and her colleagues from the Jonathan Eisen lab up at UC Davis. They noticed that soy milk had turned blue and that this tofu had turned blue. And being microbiologists, they're like, we got to take a look at this. So what they did is they aliquoted it onto an LB media, streaked it out, was still producing blue, and they isolated different strains from those. Some of them not only were blue, but they also fluoresced in response to UV light. And what they did as well is they sequenced some of the ones that were some of the colonies that they had isolated, and the two that were blue were Pseudomonas species. And so specifically when they looked more into it was Pseudomonas carnis. And so we're like, okay, most likely we have a Pseudomonas species in here. Most likely they've already identified what it is. And so these pseudomonads, they produce a compound called pyocyanin. And so that's what the blue is from that. So we're like, okay, it's maybe that, but why not? You know, it could be something new. It could be a different version of it. It could have a different application. It could be easier to produce for some reason. So let's just take a look. So I'm a microbial genomicist, so I study the genomes of microbes, specifically microbial communities. So I'll look at what's called the metagenome. The metagenome is you take the DNA from an entire microbial community, you sequence it and see what's in there. So what I did is I isolated the DNA from one of those swabs. And so this is a spectrometer curve where DNA absorbs at 260 right here, and so it shows that I did isolate the DNA at a pretty good yield. And Seacoast Genomics, which is a company out in New Hampshire, they were like, gimme, let me sequence it. So we're all like, sequence all the things. So I sent it off for sequencing to them. While also, so this is what the actual like isolated DNA looks like. It's just a little clear bullet of liquid. So I sent a bullet of that to Sebastian, one of the 8 swabs he made me dig into. And then I also made this media from like rich bacterial media with beef bouillon put into it, because I was like, why not? Maybe it grows in that. And it was kind of funny. It smells like top ramen. When I actually make that, I'm like, this is a little weird. I'm like hungry all of a sudden. But so meanwhile, I sent all these off to Sebastian as well so he could do his thing. Because his home lab is outfitted for culture work, he went and did his thing. And so the first thing he did is he took that swab and he resuspended it in rich bacterial media. Kind of like let it suspend so it's like nice and turbid, so some cells and came off of it. And then he plated a little bit of that media on 2 different types of media. One, which promotes these pyocyanins to turn blue, and another one that promotes fluorescin to be produced, which causes them to fluoresce. And so what was also funny on top of this, when this goes, come on. After he had plated those, he took that tube and he spun it down. And so he spun it down and he saw that the pellet, so the cells spun down at the bottom and the pellet was still blue, but the media, you can't see it here, was also still tinted blue. So both the pellet and the media were tinted blue. Well, first, he's like, I resuspended from a domestic Q-tip. And Mike pointed out, I appreciate the clarity that it was a domestic Q-tip, not a Q-tip that was caught in the wild. As scientists, we have to be exact, right? So then Michelle chimes in with, this makes her sure that this is not probably a pseudomonad producing pyocyanin, because she's worked with those in the past. And usually while the media is blue, the cell pellet still stays like this like beigeish pinkish colony that way. And so, so she's like, most likely it's not the culprit. So the soup plot thickens. But Philip, I got your puns for you. So Philip chimes in, well, it is like a soup, you know, you might have like some veggies and some debris and stuff that would come down with it. Like maybe the blue is from like the other stuff in the soup. So then Sebastian comes back with, no, I trust biology's like, I'm biology. I trust Elinne's like culture instincts and her techniques and stuff. And I'm like, wait. I literally dumped my hand with Q-tips into a bag that was sitting in the trash for 2 days. So like all bets are off on this one, but he was right. I definitely didn't get any other plant material. It was really just the supernatant of the broth, because no matter what, still a microbiologist at heart. So, but I show you this because this is really the way that scientists banter when it comes to like this type of stuff. It seems kind of ridiculous, but it's true, because you got to get your materials and methods in order. So I said, yeah, all bets are off on this one. I was literally dunking Q-tips into the soup in a double trash bag while gagging. But I was just thinking, like, how absolutely ridiculous when we write this up in a paper, the materials and methods, and the acknowledgments section of the paper is going to be. And then lastly, we figured out how the recipe will appear for the blue soup paper. Recipe available upon reasonable request. So this is code. If you've ever been involved in scientific publishing at all, that's basically code for we're not giving you this information unless, yeah. So hence it's going to be a very protected soup recipe. But it's also a joke because everyone involved in this is really, really in favor of open science and being really transparent with everything that we do and make it publicly accessible. So Sebastian's plates grow, and the one that had the media that encouraged the expression of the fluorescence, they did fluoresce. So we're like, cool, you know, you have something that's something that's growing on there that does fluoresce. But then the one that was supposed to secrete cause the pyocyanin to secrete didn't happen, so nothing turned blue on that one. Meanwhile, I also took a swab and I had streaked it onto the media, those top ramen media, and I put it at different temperatures. This is like body temperature, and then this is room temperature, and this is refrigerated temperatures. And what was wild was that like the different microbes grew at the different temperatures, which is expected, but also how well the stuff in refrigerated temperatures grew. But still, no blue. There's still no blue here. And we're like, oh, okay, fine. So then Sebastian's like, okay, let me try something else. Come on. Half the time it works, half the time it doesn't. So he read that some Pseudomonas fluorescens species can turn soft cheese blue. And so what he did was he had these mozzarella balls, and he put them into these well plates and then aliquoted a little bit of the different cultures from the different like strains that he isolated onto there to see if any of them would turn blue. This is my favorite thing. He labeled this "science cheese do not eat." You gotta make sure, you know, because he has a home lab. You wanna make sure that his mom doesn't come in and be like, ooh, yum, you know, type of thing. So, Sebastian's little quips on all this have just been freaking hilarious. But a few days later, 3 days later, the ones that were at 30 degrees Celsius they smelled unique and they melted a little bit. They fluoresced, but there was still no blue. So it was like, okay, cool, we got a funky cheese experiment, got some fluorescence, but still no blue. So we're scratching our heads about this, but we get busy because Sebastian has a day job aside from going after blue soup. So do I, but I was down with COVID during this time, so I was also isolated for a week. So we were just kind of like off taking a break. But then Sebastian looks in his fridge. We're like, everyone, keep calm. Turned bright blue. Was more like an indigo, really, but after like another week, the streak plates that were in the fridge turned blue. We're like, oh my God, we finally got it. And so I was like, well, I need to get back out of isolation So I can go check the ones in my fridge, and the ones in my fridge also turn the same indigo color, even though it was on different media than what he was plating. And so what was interesting was that the ones that were at refrigerated temperatures at 4 degrees Celsius were blue, but the ones at room temperature were not. And so we're like, this is really cool. This is definitely something that's specific to it being in the cold. So we're like, ooh, this is exciting. And so Sebastian then there were some strains, you can see a couple colonies. This is dark blue. This one's dark blue, and there's another one up here that's dark blue. He picked those and he restreaked those so that we could sequence those later. So while we're getting excited about the blue, we have 2 types of sequencing data that come back. One is called 16S sequencing. That's when you sequence a single gene in a population of cells, and it kind of just tells you who is there and in what relative amounts. That's really the only information you can get from that. And then we did another type of sequencing called shotgun sequencing, which has that name because you sequence everything, all the DNA, so all the genes in a population, every bit of DNA that's in there. So the 16S-seq, it's faster, it's cheaper, so that's why we did it first real fast. And we've preliminary look at it. It's not surprising that the vast majority of the population is Pseudomonas. There's different Pseudomonads in there. We're like, okay, so probably is Pseudomonas. But there's a chunk of Enterobacter and Lactobacilli in there too. And then we get the shotgun results back, and the preliminary analyses support the 16S data, that it's predominantly Pseudomonads, but we're gonna dive in. But one thing I was particularly proud of is despite using the domestic Q-tip and my crude sampling techniques, there was only 0.08% of human DNA in there. So I was like, cool, all right, cool. So I still got it, right? I'm kidding. So then I got really busy, and SeqCoast were like, can't sit on our hands anymore. Come on, we got to analyze this stuff. We got to know. We have data right in front of us. We need to analyze it. So they couldn't wait for me to look into the shotgun data, so they went ahead and did the analyses themselves. And so what they did is they had the shotgun DNA sequencing. What you first do is imagine a metagenome, like a microbial community, is you have 1,000 jigsaw puzzles, and each of them are 1,000 pieces each. The numbers are way bigger than this, but I'm just rounding it up so that you can kind of imagine it in your head. What you do to sequence is you isolate everything and you undo all the puzzles. So you have a bunch of puzzle pieces from 1,000 different jigsaw puzzles and 1,000 pieces per puzzle. So that's what you end up sequencing. And then you have to use computers to assemble those puzzles back together. So first, what you would do is you just look at your pile of puzzle pieces and you try to find some connections. So you try to build them like into as many kind of clumps. Like you have one that's like a thing of seagulls. Okay, let me build as many seagulls as I can. You have another one that's mountains. Okay, let me build as many mountains as I can. But they all have sky and they all have like maybe grass or something. So you can kind of put them together into chunks as best as you can. But then you look at a database. Your database is your covers of all those puzzles. And you're like, okay, this chunk kind of goes there. This chunk kind of goes there. But also you kind of want to put them into bins based off of how similar they are. So you're going to put all the seagullish ones that are even though things are left over into one bin, and you kind of put them in. So you use these combination of techniques to kind of assemble your puzzles and put them into different bins as you will. So you can separate those into 1,000 different puzzles. So as you can imagine, it's not a perfect system, but you can get kind of most of those puzzles kind of put into those bins depending. So they did this, and then they found that one of the puzzle pieces was, one of the puzzles they assembled was Serratia quinovorans. And so I say Serratia, I did a Twitter poll, and the majority of people said it's pronounced Serratia instead of Serratia, so I apologize to the non-US folks. That is what the Twitter poll said. So it is Serratia quinovorans, and in that puzzle, there is genes that encode the biosynthetic pathway for indigoidine. And so most likely what this is that the blue that's being produced is indigoidine, which is used as an environmentally friendly dye. And so we're like, cool, so our sequencing data suggest in that bin that Serratia quinovorans, which only makes up 8% of the population, it's not the majority of those pseudomonads. So 8% of the population are these Serratia quinovorans, and most likely it's indigoidine that is actually producing this blue color that we're seeing. So again, this is just genomic. This is looking at the genomic data. We have to validate it. So, but in the meantime, Twitter ends up doing its thing, where we thought it was a Pseudomonas species at first, and then here comes Serratia quinivorans passing it by and everything, like, oops, sorry, you're the one in charge now. Brian, wait, this one. Used to be Pseudomonas, and then now it's Serratia quinivorans. And my personal favorite is this one, the Serratia quinivorans, like now over, like being the popular one. And of course, because Twitter couldn't let it go, now you have to make it blue. So there's a lot of memes coming from this, which was like pretty shocking because everyone thought that it was going to be a pseudomonad that's in there. So what's next? The first thing we need to do is sequence more things. So we're going to sequence those strains that Sebastian had isolated that either uptook the blue or some that were producing that blue. And then we also, come on, next. Bring in the chemists. Chemists, sometimes they're good for something. Kidding, y'all. And so we did have our Department of Chemistry on campus is super, super generous. So they did take some of the swab of the blue soup and then they ran liquid chromatography mass spectrometry on it so that they can identify what compounds are in there. But also we're going to try to extract the blue from those plates and run an NMR on it, which could actually get the structure of that molecule, because maybe it's a different version of indigoidine. Maybe it's something new. It might just be the same thing, but you won't know until you check, right? And then lastly, publish. And what'd be really, really cool is that I get to be on a paper with my mom, and not just my mom, but like the scientific community as well, that like brought me so much joy throughout this whole process. And so this made some headlines. It started by blowing up on I Effing Love Science, and then other journals, Biotechnique, CNN. It's funny, my fiance is an organic chemist, and he's like, you're not even in chemistry and you made, so this is Chemistry and Engineering News. He's like, you made it before I did. Like, what the heck, type of thing. And then MIT Technology Review. It was also on NPR in Colorado or something. So it was like, I'm like, y'all are getting excited about blue soup, but it's fun. And also someone made a color palette from the blue soup. So whenever and they already are like using it to make charts and publishable figures and such. So it's a very soothing palette. I wonder if anyone can use it for like their interior decorating or something. But really what this all is about is a reminder of why we do what we do. Like, science is about curiosity. Science is about helping improve our world. Science is about working together on all of this. It's about the community that comes together and the absolute excitement and joy and the openness and communication and the willing to share ideas and expertise has just been absolutely like overwhelming from the science Twitter community. And not just from scientists, you had so many people that were not scientists that they were just curious about it. And that just reminded us why we get into this, because we're so busy chasing grants and trying to publish, and like with the job market being what it is, it's just so easy to be stressed out and forget about why we fell in love with science. And this was really a great reminder. And not only that, but Rebecca puts it perfectly. This is why I love my fellow scientists in science Twitter, because especially as someone who was a junior faculty when the pandemic hit, this community made me feel less lonely during the shutdown when we couldn't be around anyone. And they not only were there for joy and support and discourse, that's a thing, scientists do that, but they shared resources and like helped each other through it and brought so much joy through all of this that it's just been such a special part of my life, and I'm so grateful to be a part of it. So this is the story, like I said, of a microbiologist who studied her rotten soup and found and built an awesome story and community from that. That was a lot of fun. So thank you all so much for your attention, and I can't wait for where this goes.